Now we will talk about how the electrical signal triggers contraction of the muscle cell. This process is referred to as excitation-contraction coupling. We'll use this slide and the next one to describe the process. For now, just ignore lines labeled 6 through 11. You can use these later to review the entire process if you want to. Again, after acetylcholine is released, it travels across the synaptic cleft and binds to a receptor. This opens up a channel and allows sodium to enter. This triggers an electrical signal that travels across the sarcolemma and enters the T-tubules. Once in the T-tubules, the electrical signal reaches the terminal cisternae at the triad and stimulates the release of calcium from the sarcolemma. Now let's look deeper into the myofibrils and at the myofilaments. The calcium ions that were released from the sarcoplasmic reticulum will move into the myofibril and bind to the troponin proteins on the thin filament. When calcium binds to troponin, it undergoes a change in shape which causes tropomyosin to move. When tropomyosin moves, it results in the exposure of active sites on actin. Once the active site is uncovered, the myosin heads can now interact with the thin filaments. The interaction between myosin and actin creates a cross bridge. This begins the contraction cycle. Myosin is an enzyme. It's an ATPase that breaks down ATP into ADP plus P. When this happens, the ADP and P remain bound to the myosin head, which allows the binding of myosin to the active site on actin, creating the cross bridge. When the cross bridge forms, the ADP and P are released from the myosin head. As this happens, the myosin head pivots and pulls the actin toward the center of the sarcomere. This is referred to as the power stroke. The next step is cross-bridge detachment and happens when a new ATP molecule comes in and binds to the myosin head. The binding of ATP to the myosin head disrupts the linkage between the myosin head and the active sites on actin. The myosin head will break down the ATP into ADP plus P, and the process will cycle again. This process will continue as long as calcium remains bound to troponin. This is a brief overview of the sliding filament theory of muscle contraction as we looked at earlier and how it is tied to the interactions between myosin and actin that are necessary for muscle contraction. The contraction cycle requires cross-bridge cycling. Essentially, this refers to the attachment of myosin to the actin, the pivoting of the myosin to pull the actin toward the center of the sarcomere, and the detachment of myosin after the binding of ATP. As mentioned earlier, this process of cross-bridge cycling will continue as long as calcium is available to bind to troponin. Use this image to review the events of muscle contraction. Be sure you understand what's happening before you move on to the next portions of this recording. Now let's look at the steps involved in muscle relaxation. The first thing to happen is that acetylcholine is broken down by an enzyme called acetylcholine ester. Breakdown of acetylcholine will prevent it from binding to the receptors on the muscle cell membrane. This results in the closure of the sodium ion channels and an electrical signal no longer occurs. The sarcoplasmic reticulum recaptures all the calcium ions. Without calcium binding to it, troponin moves and the tropomyosin covers the active sites on actin. This will block cross-bridge formation and contraction ends. Relaxation occurs with passive return of the muscle to its normal length. The stimulation of a single muscle cell to contract is a little different from the stimulation of an entire muscle to contract. Individual muscle cells obey what is called the all or none principle. This means that when a single muscle cell is stimulated to contract, it will either contract fully or not at all. Recall when we discussed muscle tissue previously that a single muscle cell is the entire length of the muscle it is part of, and in some cases these muscle cells are long. It would not be very effective if only a portion of a single muscle cell contracted. This is why single muscle cells must obey this all or none principle. Now, stimulation of an entire muscle is different because it will involve only contraction of some muscle cells within the muscle based on the force that is required to move the bone. This is referred to as the principle of graded strength. Think about it this way. If you contract your bicep muscle to flex your forearm, do you think it would require the same strength, more strength, or less strength than it would if you flex your forearm with a 15-pound weight in your hand compared to no weight in your hand? The variation in the amount of force needed to move an object is related to the number of muscle cells that need to contract within the entire muscle to perform the movement. So while individual muscle cells must obey the all or none law, or principle, an entire muscle cell does not obey this law. This is important to understand and it means that muscles will contract in a graded fashion to provide the required force needed to move a bone and/or object. First, we will look how an individual muscle cell responds to a single stimulus. We will then build on this to understand how an entire muscle responds to a stimulus. The twitch is the response of a single muscle cell to a single stimulus. The twitch of a single cell is too brief to be part of any normal activity. We can trace the pattern of a twitch using a myogram. The myogram is divided into 3 phases. The latent phase is the shortest phase and begins with stimulation of the muscle cell. During the latent phase, the electrical signal is moving across the sarcolemma toward the T-tubule. From here, it reaches the sarcoplasmic reticulum, and calcium ions are released and enter the myofibril, where they will bind troponin. This causes exposure of active sites on actin. This leads us into the contraction phase. During the contraction phase, cross-bridge cycling is occurring and the muscle cell is shortening. Once peak tension is reached, we move into the relaxation phase. During this phase, calcium ions are moving back into the sarcoplasmic reticulum, the active sites on actin are covered, and cross-bridge formation stops. A single muscle twitch is too fast to be part of any normal activity, so we must sum them to get our muscles to contract with enough force to move something. Summation involves adding individual twitches together to produce a graded contraction. There are 2 ways to sum twitches. The first way is called multiple motor unit summation or spatial summation. This type of summation results in an increased force of contraction due to an increase in stimulus strength. In this type of summation, an increase in stimulus strength will cause an increase in the number of motor units contracting. As more motor units contract, the force of the contraction gets stronger. In this image, the top line represents the strength of the stimulus, and the bottom line demonstrates the strength of muscle contraction because of the stimulus applied. The first stimulus that results in contraction of the muscle is known as the threshold stimulus. Notice there are some stimuli that do not produce a contraction. These are called subthreshold stimuli. Notice that as the stimulus strength increases, so does the force of contraction by the muscle. This continues until a certain point is reached where the force of contraction does not increase with increasing stimulus strength. The stimulus that results in the maximal force of contraction is the maximal stimulus. All the stimuli between threshold and maximal are referred to as submaximal stimuli. The question is, why does an increase in stimulus strength increase the force of contraction? This effect is due to the recruitment of more muscle fibers and more motor units to participate in the contraction. As additional muscle fibers and motor units are recruited, the force of contraction increases. Remember, your muscles are made of multiple different motor units, and so the neurons stimulate various muscle fibers. As the number of muscle fibers contracting increases, so does the tension produced by the muscle. Now let's apply this idea of motor unit recruitment to the graph. This represents a muscle in cross-section. The white circles represent non-stimulated muscle cells, and the red circles represent stimulated muscle cells. When the first stimulus is applied and there is no contraction, this indicates that the stimulus is not strong enough to activate any of the muscle cells in that group. A second stimulus that is slightly higher was able to stimulate a single muscle cell, but it's not strong enough to initiate muscle contraction that can be measured. The third stimulus, which we labeled as the threshold stimulus, was enough to see contraction of the muscle. Notice this stimulus caused the contraction of 2 muscle cells. As we continue to apply stronger stimuli to the muscle, additional muscle cells are stimulated to contract. These additional contracting cells increase the force of contraction. Once the maximal stimulus is reached, all the muscle cells that are available within the muscle are contracting, so any additional stimuli applied cannot recruit any more muscle cells, which is why we don't see any increase in the strength of contraction. In multiple motor unit summation, the force of contraction increases by increasing the stimulus strength. With each increase in stimulus strength, more muscle cells are recruited to participate in the contraction until all the muscle cells that are available to participate are contracting. The second type of summation is called temporal wave summation or just wave summation. In this case, we're going to increase the force of contraction by increasing the frequency of stimulation. When the frequency of the stimulation increases, the next stimulus will arrive before the relaxation phase ends. If the frequency of the stimulus is fast enough, a sustained muscle contraction is reached. This sustained contraction is called tetanus. Tetanus occurs when the twitches are summed together to produce sustained muscle contraction. The difference between wave summation and multiple motor unit summation is how the stimulus is applied. In motor unit summation, the stimulus strength is increased until all the muscle fibers and motor units are recruited. In wave summation, the stimulus strength remains constant, and instead the frequency in which that stimulus is applied changes.